No, I don't say that right now.
Okay, so I'll be back in 2 minutes.
Yeah. Hello, everyone, and welcome back to the online Biological Sciences Seminar Series of TÜBİTAK Research Institute for Fundamental Sciences, which we organize for national and international audiences to mitigate the demoralization effects of COVID-19 coronavirus outbreak on scientific thoughts and science-based actions. Today is another wonderful day. As I has the honor of introducing our speaker, Professor Nir Ben-Tal from George Weiss Faculty of Life Sciences, Tel Aviv University, Israel, a well-known scientist, world-class expert in computational biology. He's going to talk all about global view of the protein universe. At the end of the talk, we will have a question-answer session where the questions can be asked by sending a message through chat button of the Zoom platform. Professor Ben-Tal research focuses mostly in general area of computational structural biology. This includes both methods developments and applications to selected problems. Professor Ben-Tal's laboratory has developed the CONSAF methodology and web server for the detection of interactions regions in proteins based on evolutionary information, jointly with Melrose and Papka laboratories. Professor Ben-Tal's laboratory has also developed methods for prediction of structure and motion in membrane proteins and studied selected proteins, including human copper transporter 1, the ATP7B copper, ATP sodium proton exchanges. Recently, the laboratory also started to conduct visual screening for drug-like compounds with selected protein targets. Professor Ben-Tal completed his bachelor degree in biology, chemistry, and physics at the Hebrew University of Jerusalem in 1988, and his Doctor of Science degree in chemistry at the Technion, Israel Institute of Technology in 1993. Later, he did his postdoctoral training in biophysical chemistry at Columbia University, New York. In 1997, he accepted a faculty position at the Department of Biochemistry and Molecular Biology, Tel Aviv University, where became full professor in 2007. Professor Ben-Tal is an associate editor of the journal PLOS Computational Biology and a member of the board of reviewing editors of eLife. He also was a member of the editorial board of the Journal of Biological Chemistry. He's a co-author of the well-known textbook, Introduction to Proteins, Structure, Function, and Motion. Professor Ben-Tal has many honors, amongst them Award of Excellence of the Wolf Foundation's Israel, Award of Excellence of the Teva Foundation's Israel, Kazan Chair of Structural Biology, Tel Aviv University, the NATO Science for Peace and Security Prize, jointly with Oded Levinson from Teknion, Israel, and a Turkish professor, Türkan Haliloğlu from Boğaziçi University. With this, I want to hand over the word to Professor Ben-Tal. Nir, thank you once again for joining us for this seminar, and please begin your talk.
Thank you very much, Ali Kram, for the very kind introduction. Let me just share my screen with you. For it is to be okay. Yeah. Okay, I hope that I'm...
yes, it's quite not nice.
Thank you very much.
Okay.
So again, thank you for the invitation, and thank you also to my friend and colleague Türkan Haliloğlu for suggesting me for this a very interesting opportunity to talk to you all. And I'll describe our studies on global view of the protein universe. It's a joint work with Sergei Nepomniachtchi, Aya Narunsky, Ron Solan, Mit Tessel, Rachel Kolodny, and several others that, that you will see during the talk. And the idea is to try to figure out what is it in current protein universe that can teach us something about the evolution of proteins. So nowadays, proteins play central roles in living organisms, and they are involved in all the key functions of the cell, in metabolism, in DNA replication, in motion, signaling, etc. And because of how much they are entangled with other things, it's really difficult to imagine how life could have emerged without proteins. So in a sense, when we study how proteins emerged, it's very tightly linked to how life itself emerged. Now, when we try to decipher how proteins emerge, there are various conundrums that are interfering with our thinking, and one of them is demonstrated here. So proteins are coded by DNA, but DNA itself is synthesized by proteins. Therefore, it's really difficult to tell which one was before. We have a chicken and egg kind of problem. But really, we want to know, as we want to know, as some of us want to know how the universe emerged, I care about how proteins emerged. And there are several ways to go about it. One approach, which I will not be dealing with here, is the prebiotic chemistry approach, where you think about atoms and seeing how they combine to form small compounds and then amino acids and the amino acids polymerize to form peptides and eventually proteins. And a lot of you probably remember the Miller experiment in this direction, but it's a whole lively community that is very interesting. Here, I'll take a complementary view, looking at it from the other end. It's an archaeology-like approach where we are going to use current protein repertoire and try to use it to speculate about how proteins emerged and evolved. And this is like an illustration of how protein might have evolved. So it started with some protein sequences, amino acid sequences of the protein. And as time proceeds, because of various evolutionary processes, a whole selection of protein emerged. However, of course, a lot of them were structurally unstable and couldn't take it. The biophysics made them. They got deleted because of biophysical restraints. And please don't take it as proportion. I mean, the real fraction of those that didn't make it is much larger than what we see here. Later, there is also a selection based on function. I mean, evolution goes by function, not necessarily by structural stability. So a lot of them got deleted because they were not functional. And then, oh, I'm sorry, I'm confused by the Zoom. And eventually, today, we end up with a set of protein, which again, some of them got deleted because they are not stable or not functional. And from those that we know now, for some of them, for a very minor fraction of them, we know their three-dimensional structure. And for some of them, we also know their function. But for the vast majority, we just have the sequence. And we don't know all the history. We just see contemporary proteins, and the idea is to try to use this information and dig back and answer two questions that are related. One of them is how proteins evolve, and one of them is how proteins emerged. What is it that started the proteins? So hopefully, by studies of the protein universe, of the current protein repertoire, we can address both. Now notice that, well, first of all, just to lower your expectations, don't expect to really understand all this in the end of the lecture. I still don't know, and I hope to eventually know it within my lifetime, but then also don't leave, because I believe that what I will say is interesting enough and provide at least some hints towards those questions. Now, how proteins emerge, meaning that I have to go really, really back in time, maybe 4 billion years ago, when proteins are assumed to have started, and that's really, really difficult. How proteins evolved can change. So, for example, if I just care about the proteins that have been here maybe 3 years ago, or you know, several hundred or 1,000 years ago, that's relatively easy. But if the further I'm going back from what we have now, the more difficult the question becomes. So how proteins evolve, that again depends on how long ago I dare to look back. This is an outline of my talk. So it started with the introduction. I will present a global view of the protein universe and what we can learn from that. I will briefly introduce a tool that we felt we had to devise along the way. I'll do a detour to just to show what we can do about the evolution of a group of proteins called outer membrane beta barrels that are shown here. And I will show how we suggest to expand the known architecture of those proteins. I will then try to think about the elementary unit of the protein evolution. And eventually I'll talk about how a certain function evolved, and that's the function of adenine binding. So our study is about the protein universe. So again, we start with the current proteins, and it's easy usually to start with proteins of known structure. So we usually start with those. And of course, we can't use all of them, so we just sample some of them. Of course, we are not the only ones who do it. Others have been doing it, and this is reviewed, for example, here in this article that we wrote. So there you'll have reference to many others who work on this field. In my talk, I will almost not refer to previous works by others, that is. But you can find a lot of references here. I will introduce ideas that others have been framing. So when presenting the protein universe, and again, I'm summarizing here work by many others, there are three things one needs to consider. One of them is the entity to be used. For example, are we going to use the full protein chain or maybe some parts of it? For example, domains is a very common choice. The next thing is the measure of similarity between the entities. And common choices are sequence or structural similarity, sometimes both. And then also whether it's going to be global over the entire protein or based on some of them, some part of the entity. And also whether I want to show similarity among all the proteins or just some of them. Finally, there is the representation, and the most common choice is least, but there are other possibilities. So, as I said, the most common entity to be used is our domains. I'll say some more about domains, but these are parts that we see again and again in various proteins, which is why people focus on domains. And again, the most popular way to present protein space is based on lists. So people have developed domain databases, for example, CAS, SCOP, ECODE, and these are very useful. You can see here domains, and you can see that some of them are structurally very similar, and very often they are similar in their sequence. And those domain databases, those lists, they are very useful when we care to know about proteins that are very similar to each other. There, we don't need much more than just this. However, if we want to know some more general properties of proteins, sometimes it's more useful to use other means of presentation. For example, it's possible to present all the domains as points in two-dimensional or three-dimensional space. Here, I will use graphs. And in the first part of the talk, I'll present studies that started maybe 7 or 8 years ago, where we started with scope domains, maybe close to 10,000 of those. And we looked for similarity, for local similarity in sequence and structure, and presented the result using graph. And I'll show what I mean by that. So each of these entities, here and here and here, we have is a domain. So we have 4 of those, just as a demonstration. And the slide shows that certain regions are shared between these pairs of domains. So the bluish part is shared between these two and the brownish between these two, and the greenish between those two. And then it's possible to, and here are the exact details. So they are structurally similar over 68 residues with 30% sequence similarity, and you can see it for the other things. And then we present it as a graph, as a network, where each entity is a domain, and they are linked by an edge if they share some similarity between each other. And how to detect similarity, that's arbitrary. So here, for example, the threshold was that at least 75 amino acids should be similar, and they should be superimposed structurally within 2.5 angstrom, and they should have 30% sequence similarity. But there are other legitimate choices, and we don't know ahead of time which makes sense. So we tried several, and depending on the choice, we get different networks. So when the threshold is very lax, then everything is connected. We see almost a single connected component and very few isolated islands. And of course, if I used more lax threshold, everything would be connected. On the other hand, when I use more strict threshold, I essentially lose the network. Everything is just isolated islands, or even just specific domains that are disconnected from each other. And I can think of it actually in terms of evolutionary process. So as stricter I am in terms of the threshold, the more recent evolutionary event I trace. So this is very lax, and more strict and more strict. And eventually I get to those proteins that are very, very, very similar to each other. And it's interesting, I would argue, to look at this process and to think about it. But also, we like to determine a certain picture of the protein universe, and we have to make a somewhat arbitrary choice for that. Our choice was based on how well we can decompose scope folds into their proper annotation. So how many times we mix unrelated scope folds in the same cluster versus how many times we can find related scope folds in the same cluster. And ideally, we should have been in this corner if everything went perfect, but it's not. So, and here are various combinations of threshold that we tried. The closest to the corner is this specific choice, and I'm going to present results obtained this way. However, the qualitative conclusion that I'll make are valid also for other choices. And you can see that really protein space include large connected component as well, or living side by various isolated islands. This goes back to a very old question in protein studies, in whether protein space is continuous or discrete. This is not a well-defined mathematical question this way. And here with the graph representation, we get a quantified way, a formal way to ask it. And what we see is that protein space features both a discrete region of isolated islands, as well as continuous region, where you see a lot of domains that are connected to each other. In this picture, they are colored by their secondary structure elements, alpha helices, beta strands, and mixtures, but it's not particularly relevant for us. The most important thing is that there is a continuous and discrete region of protein space. The subdivision between this connected component and isolated island changes depending on the exact threshold, but I will always find them living next to each other. Now, what are the isolated islands? And I can show it, but I can reveal it by coloring it based on a different color scheme, and that's based on scope families. And you can see that each of the isolated island is really a family of protein. For example, the blue island here is a globin-like proteins, like hemoglobin, myoglobin, that they are similar to each other, and they are an isolated group. Likewise, each of the islands, I didn't color all of them, but each of the islands correspond to a specific family of protein. For those regions, the scope definition, the scope hierarchy works perfectly well for those isolated islands. However, we can see also in the large connected component, a region where, again, we see specific families of domains that are right next to each other, like neighborhoods, but they are entangled with each other. They are very well mixed. If you want, it's like neighborhoods in Istanbul that are very crowded and very packed to each other. And whereas the isolated islands is like a rural area of the type of Eastern Istanbul, like with the isolated villages, or at least they used to be isolated maybe 30 years ago when I hiked there and enjoyed. So for this region, the large connected component, the scope representation or any representation using a list and hierarchy, they are not very helpful because it's difficult to discriminate between the various protein families, so-called. It's all a big mix. I will argue that from an evolutionary point of view, this region is actually the most interesting because that's where you can easily move between very different proteins, between very different domains. And I will show you that these domains have different shapes. So for example, you can think of the collection of edges here as evolutionary pathways. And I'm showing one of them that shift between 8 different domains. You see that they are very different in their structure. And of course, they are also very different in their sequence, but they do have enough in common so that they are linked. And you can see that they are also from very different types of folds, like FAD binding domains, nucleotide binding domains, thin barrels, flavodoxin, etc. By the way, all of these are very ancient folds. So, and it's encouraging that they are found in this large connected component. Along the way, in order to connect the network view to the structural view, we had to develop a tool. So presenting the network, we used Cytoscape, which is a tool that very often biologists use to present large data that can be represented as networks. And then there is another tool, molecular viewers like PyMol, etc., that are used to view molecules, to view proteins. However, we wanted to seamlessly go between these two representations, and then we developed Cytostruct, which is, and we thought that it can be useful for others, so we published it, and of course we made it available. It's just a plugin or an app for Cytostruct, and we give it away as in Bitbucket, etc. With that, I'm going to make a small detour just to show you what we can learn based on these tools and principles for one specific case, and that is beta barrels. These are membrane proteins. You can see roughly their shape, and in collaboration with Joanna Slutsky, we studied their evolution, and I'm summarizing a very like 2 papers here, but eventually the conclusions are that 2 of them are separate families that don't share origin with the prototypical outer membrane beta barrels. I will focus here on the outer membrane beta barrels, and the point there was that we showed that for the we and others, that for the most part, for example, Andrei Lupash, that for the most part, they evolved by adding more of those beta strands so that the barrel increases, and then it increases from 8-stranded to 14 and then to 22 just by adding beta barrels, beta strands. So, but while doing it, we were wondering if there are more architectures beyond those that we can see, beyond those that we know. So the evolutionary analysis showed us that just we add beta hairpins and obtain barrels of greater diameters. And actually, there was a study that showed experimentally that when you link 2 8-stranded outer membrane proteins to each other, you form a single large barrel with 18 strands. And we were wondering if that's the only possibility. What if it's also possible for the 2 barrels to stay next to each other without combining to a larger barrel? And for that, we created a set of single barrel proteins of known structure. We represented them as hidden Markov models, which is some technique. For this talk, it's not important exactly what it is. I can answer questions, but it's important that for all of them, the structure was known, so we knew the number of strands, and then we combined them with a lot of other sequences of unknown structure, but that were similar enough. And then we looked for cases where in the same sequence, we find concatenation of 2 single barrels. And that's the result. That's a phylogenetic tree that tries to capture the relationship between those multi-barrels that were detected. For example, this one here means that we detected a pair of 8-stranded barrels connected to each other on the same sequence, and that's in cyanobacteria. Cyanobacteria also have 3 8 barrels tagged to each other and 12-12 and so forth. So we can see many combinations, including here a combination of 11 8 barrels concatenated to each other. So recall that this is just a prediction, and hopefully, eventually, we will know the real 3-dimensional structure of some of these to validate it completely. But what can we do computationally for validation before we know the structure? So to try to get at it, we looked at co-evolutionary pattern. So in a beta barrel, we see strands that are connected to each other, right next to each other. You see 2 consecutive strands, and here is a third. And if we change one amino acid, then another amino acid that in direct contact with it may have to change in order to tolerate the change. And with that, we can detect in the sequence a correlation between these 2 positions. So there are methods, and in particular, the Raptor server from Jimbo Zhu groups and also others, to detect these correlations in sequence. So for that, we usually need many proteins that are similar to each other or that share the same evolutionary origin. We call them homologs. And in this case, I showed an example of one case where we managed to find almost 2,000 such homologous proteins. And this is the kind of pattern that we see. So this is the sequence versus sequence, and we look for correlation between, let's say, amino acid number 1 and itself, or amino acid 1 versus the amino acid 400 and something. And we see that there are very few co-evolving pairs of amino acids, and they are along those diagonals here and here. And what are these diagonals? So these diagonals correspond to beta strands that are next to each other and interact with each other. So look at this example. That's one strand, and it should interact with the one next to it. That would lead to this pattern. So this is a predicted contact based on the co-evolutionary pattern. So this would correspond to, theoretically, it would correspond to predicted contact between strand 1 and 2, 2 and 3, 3 and 4, and so forth. And if you count, there are 7. So that would correspond to contact between the 7th and the 8th. And even better, we see here a pattern that would correspond to the closure of the barrel. So that would be contact between strand 8 and the first strand to form something that looks like this barrel with 8 strands. You see, that's the last and that's the first, and they should also co-evolve because they are in contact. So we have this. Likewise, you can see here pattern that would correspond to another barrel, and it's a continuation along the sequence. More importantly, we can see, I mean, the alternative evolutionary scenario would be for those barrels to form one large 18-stranded barrel. But that would be in conflict with these missing links. So there is hardly any coevolution here between those putative strands, and also there is no signal for closing of a 16-stranded barrel. So that's another validation of the formation of 2 barrels, 2 independently folded barrels. Okay, with this detour, I'm going back to the main theme, and I'm returning to this view of the protein universe, and recall that this is all when we started from domains. The reason that we started from domains was that because they were suggested to be an elementary evolutionary unit. There are evidence that they can fold autonomously, and that they can evolve, that they can undergo mutations and change their function, undergo slight structural changes and mechanistic changes. So that's a good place for safe evolution. However, one wonders if these are really those proteins, those domains as defined, are they really the evolutionary units? And maybe there are smaller units. And anyway, since domains are large relatively, how did domains themselves emerge? So while there are certain examples that domains really fold autonomously and so forth, in practice, the entities are classified automatically based on repetition. We just see them again and again, and then we assume that they are independent domains. These are the 3 main domains databases. By the way, they are not very similar to each other. The entities are basically defined based on reuse signal. And they have very similar lengths, on average 100 amino acids, but it's surprising because they, as I said, they are very different from each other. Their boundary difference, and up to 60% of Cas domains, for example, do not even have a scope and a counterpart, and vice versa. So we wanted to systematically study reuse in protein space to figure out what is really lengths of the evolutionary unit. And I'm going to present my ideas about it based on this funny example of a lamb domain and a cat domain. And let's assume that my protein space includes 3 lamb domains and 5 cat domains. And then I arbitrarily separate them to smaller units. And I try to see the level of reuse in protein space, but reuse doesn't change when I chop to smaller pieces because there is no similarity between any part of the lamb and the cat. Like the tail cat, it doesn't resemble anything of the lamb. However, if I started from Lego parts and build my lamb and cat from these, there certainly is a reuse, and the overall reuse increases as the pieces get smaller and smaller, shorter and shorter. So we study reuse in protein space, and I see that I'm running out of time here, so I'll skip the part and just say that it's basically we zoom in to the similarities that comprise the graph that I showed before, and see what it is made of in terms of similarity between the sequences of the proteins. And then with that, we can find parts that are similar. We call them themes because they are shared by several proteins. And eventually, in this case, I detect 2 different themes that do not overlap. And then I can manipulate them and find a lot of them. So for example, I can ask for themes of 35 amino acid long, and I can see this collection. So this is a theme with several, we call them theme because they come in different flavors, in different variations. So here is a theme that I detected 7 different variations in different domains. And here is a theme that has 5 different variations, and so forth. Similarly, I can look for themes of 70 amino acids, so they are longer. But then I can also discuss the size of the theme. So how many I see of the same size. For example, here I see a theme of size 5 and another one, so I see twice. And then a theme of size 2, I see 4 times such themes. So I can plot that. And that's the kind of graph that I get when I start from the ECOD database of domains. And that's the graph that I get from starting from the PDB database of full proteins. So what I plot here is on the x-axis is the theme size, and on the y-axis, the number of themes that are found. And I can see that when the themes are short, maybe two times, then I can see a lot of them. And when they are small. And when they are large, I see very few of them. So this is a power law or a power law-like distribution, which essentially everything in nature distributes this way, and so it is also for the themes. For me, what I care most is this part, which includes full proteins. And I was curious about domains. Recall that domains have a size of 100. And I was wondering whether 100 will be any different than any other lengths. And we can see that it's not. It's very similar. I mean, reuse in general increases as the theme size, as the theme lengths get shorter. So looking for 35 amino acid, I see more often than 200. However, 100 doesn't stand out. So that means that there's nothing holy about domains to indicate that they are necessarily the elementary evolutionary unit. there's a part here that measures the reuse, but I'll skip it because I want to proceed to cover more topics. I will present here a specific case and present themes on that case. This is a propeller. This is a 7-blade propeller, a type of domain that is common among proteins. And what you see here are themes that are found in this propeller. And you see in red themes that essentially, these are thick themes that cover essentially almost the entire propeller structure. However, I can look for shorter themes. For example, if I look for the green themes, all of those in the green box, there are many more of those, maybe 30, and they only share half a propeller, 4 blades. Interestingly, here, while most of them are 7-blade propellers, some of them have 8 blades. So if you want, this is an interesting evolutionary junction that connects, that relates to the evolution of the propeller and the shift between 7 and 8 blade propeller. Of course, I don't know the directionality. I don't know if it started from 7 and ended with 8 or the other way around, but it's an interesting evolutionary junction. As I look for themes that are shorter, I can see themes that are shared between more remote homologs. And then in the purple box, I see themes that correspond to only 1 or 2 blades of the propeller, and they are shared by this entire group of domains. So that's a Russian doll-like pattern that is common in evolution. It makes sense. And this is a major discrimination between our segregation into themes and the domain segregation. In domain segregation, each amino acid can belong only to one theme or another, or only to one domain or another, sorry. But in themes, the same amino acid can be a part of a short theme shared by a lot of proteins, as well as by a very long theme shared by very few proteins. At the time, I was very excited about it, showed it to my friend Turkan Haliloglu from Istanbul, and it reminded her, this Russian doll pattern reminded her how modes of motion in protein are detected, and she tried to correlate themes with modes of motion. So she used elastic network models and demonstrated it in several domains. I'm showing here a demonstration on the exact same propeller structure that I just described here. So you can see here a list of all the themes. So this is the amino acid sequence, just from 1 to 400 and something. And each of those green lines is just a theme. And you can see how there are boundaries between them, but also a lot of overlap. And below, in red and blue, you can see the 7 slowest modes of motion that control the main functional dynamics of the protein. You know that protein have to undergo functional dynamics to function, for example, to shift between active and inactive conformation of an enzyme or closed and open state of channel, etc. So this is the domain, the composition into the dynamic domains, and you can see that they correspond nicely. Here are specific examples. So this is the 5th, and this is the 7th slowest mode in this case, and you can see the representation along the sequence with relevant themes or themes or theme combinations. And here is a projection of this on the 3-dimensional structure. You see in blue one region of the domain that move in one direction, and in red, a region that moves in the opposite direction, so that there is kind of motion relative to each other. That's one of the dynamic motions. You can see also in the seventh mode. So that suggests, perhaps, that not only the themes, when they mix and match to evolve new domains, not only that they have to match geometrically, but they also have to match dynamically to form a functional protein. I'm back with this figure of the protein universe, and I'm going to ask another question now based on this. So most of those edges connect domains that are very similar to each other. But I wonder if we can find themes that bridge between evolutionarily distinct domains. That's a difficult question to answer. And first we need to understand or to define what is it an evolutionary distinct domain. And for our definition, we decided to use sequence and structure. So we decided to look for a certain theme. You see the variations of this theme, and we want to detect the same theme on two domains, domain 1 and domain 2. You see that the same theme exists here with different variations. So here there is similarity. However, we wanted to make sure that overall the structure of this domain 1 and domain 2, they are very dissimilar from each other, and also the sequence before and after the theme are very different. So they are not similar here and non-similar here. And with that, we came up with a list of bridging themes that can bridge between evolutionary distinct domains. And you can see that they are similar in sequence, but this is a measure of their similarity in structure. If they were all similar, they were in this region. This is the root mean square deviation. And you can see that actually a lot of them are very dissimilar. In fact, there is, if you want, two distributions. One of them is for themes that are very similar to each other, where the variations are very similar to each other in the same theme. And these ones, this peak, correspond to cases where the variations are very dissimilar from each other in their structure. So that's an interesting thing. Maybe a lot of those bridging themes, they undergo structural conformational changes upon shifting between the different environment, between one type of domain to another type of domain. This is a network representation of this. And the only thing that matters is that a lot of those domains that are found here are known to be ancient for other considerations. And then it's interesting that we find bridging themes between those ancient domains. I will show one example, and that's one domain with a theme shown in purple, and that's another domain where the theme, the equivalent theme, is shown in blue. This is a superimposition of those themes. You can see that each of those themes in its respective domain is engaged in binding of iron-sulfur cluster. That's a ligand. It's used to extract energy. I will say a bit more about it. Interestingly, when we superimpose the themes on each other, the two variations of the theme on each other, we see here that the iron-sulfur clusters also superimpose on each other very, very nicely. Iron-sulfur clusters are actually very, very ancient ligand, and it's considered to be a possible source of energy in the ancient history before the atmosphere changed to oxidizing. So that's for anaerobic breathing, if you want, at the time. So the fact that these two themes, or these two variations of the theme, are found in two very ancient proteins, and they bind this ancient iron sulfur cluster, suggests that it could be a very ancient theme that could be one of the beginnings of the protein universe. Sorry. Okay, there's another part which will be like 5 minutes about the emerging of adenine binding, but maybe I'm exceeding my time, so please let me know if I should proceed, or I can just quit here.
No, please continue. It's up to you.
Okay, thank you. So just a few more slides. So recall that I introduced this slide to ask about the emergence of proteins, but protein emerges for function, and this is an effort to understand something about the emergence of some kind of function, which is a binding of adenine. So adenine is also an ancient ligand. This is this picture shows the chemistry. It has a lot of hydrogen bonding donors and acceptors, and it's found in a lot of key ligands, in ATP, in DNA, in adenosine, in many other important cofactors that facilitate catalysis. So it's considered a key and an ancient ligand, and we wondered what can we say, if at all, about the evolution of adenine binding, how proteins learn to bind adenine. So adenine has 3 edges. One of them is the famous Hookston-Crick edge, which is used for pairing in the double helix. There is also the Hookston edge and a sugar edge. And previous studies that looked at adenine binding, they focused only on binding through the Watson-Crick edge. We were surprised that there is no binding through the other edges, and we went to look at it and decide. And therefore, we developed a software tool called Common Binding or Combined, which collect all proteins of known structures that feature adenine binding, and then it aligns the adenine part. Notice that adenine is found within different contexts depending on the ligand, but we align only the adenine, which is rigid. It's actually also flat, which makes it easier to present the result, but the tool can handle any kind of ligand. So we align the adenine and drag with each adenine the protein. So we end up with getting a view of the protein adenine binding from the point of view of the adenine. And then we extract the atoms using the arpeggio. We extract the atoms that hydrogen bond to the fragment, to the adenine binding fragment. And with that, that's the kind of picture that we get. So we see here the adenine and a bunch of them that are superimposed. We see that they superimpose nicely. And then we see in red and blue spheres, we see oxygen and nitrogen atoms. And in the 3-dimensional plus signs, we see water molecules that mediate the binding. And we see that as we anticipated, not only the Watson-Crick edge is used for binding, but also the other edges. Now, not all proteins mediate binding through all 3 edges. Usually only the binding is mediated through one edge. But you can see, because we see various proteins that bind through different edges, we can understand that adenine binding emerged more than once in evolution. The next thing we did was try to correlate themes with adenine binding. And we can see here mapping of themes on domains that bind adenine. And you see that the adenine bind very close to specific themes. And from that, we analyzed various themes that bind adenine, and again, we use this network, where in this case, the nodes are protein adenine complexes of known structure, and the edges connect proteins that share adenine binding theme. And we see clusters, and really each cluster corresponds to a specific binding mode. So for example, this cluster number 1, it corresponds to binding mode that used to be, it's already a known one. It's called the direct motif that binds through the Watson-Crick motif. In another one, there is the so-called reverse motif. And of course, we find a lot more. I'm not going to outline all of them. This is an interesting case because adenine binding is mediated through a combination of 2 themes, one in purple and one in pink. With that, anytime that we see a theme, we can detect adenine binding. And this is the summary of my talk. So we study protein universe to understand protein evolution. I showed you an example of outer membrane beta-barrel evolution. I showed you how reuse is in general very prevalent at all theme lengths, not necessarily of domains, but also below and more significantly, and below and above the domain size. Below the domain size is particularly interesting because it may give us hint about the early days of evolution. Adenine binding emerged more than once in evolution, and I showed you that themes are very often related to, or not very often, but at least in some cases are related to protein function and dynamics, and therefore it's possible maybe to predict function based on themes. And it's also possible perhaps to use those themes in protein design. So for example, if I have a protein and I want to make it bind adenine, then maybe I want to bring in an adenine binding theme into the protein. And now my final slide of thanks to the main collaborators, and you can see who they are. I'll be happy to take questions if there are.
So thank you very much for this very exciting and comprehensive talk.
So
maybe questions, we can take some questions.
And how do I do it?
They will come a message through chat button. So questions, please. So, Türkan Hocam, do you have some questions?
Hello, Nir.
Questions or comments, please. Questions or comments?
Okay, thank you very much, Nir, for this nice and very inspiring talk. I'm very fascinated with the themes already. So I hope that we'll get more insights with the dynamic analysis Do you think that you mentioned about the bridging you mentioned about the bridging themes. So do you think that it would be possible somehow to relate this with also the dynamics?
Yeah, in principle, yes. We need to think about how. I mean, there are themes. There are just themes as you saw before. The only thing unique about them is that they are just detected between very evolutionarily remote domains. For example, Rosman and Pilou, or Rosman and Tim Beryl, that are considered, each of these folds is considered like an autonomous folding unit, if you want. And nevertheless, we see evolutionary links between them. So to analyze, for comparison, you just need to map the dynamics to both those types of domains. But I believe that you will see very similar patterns.
Okay, thank you. And also about the structurally dissimilar teams. So they are larger than those that you originally studied, and those that are dissimilar. You mentioned about the conformational changes of teams.
No, not necessarily. I mean, we are actually looking into it now, but from my just by brief looking, they are not necessarily longer than others. So they are, on average, all of those bridging themes are on average 40 amino acids, and some of them undergo structural changes, some of them not so, and no strong relationship to the length.
Okay, thank you. Thank you.
Thank you.
Any other questions? May I ask one question? You mentioned about a design of the protein, perhaps with some properties, controlled properties, perhaps. Is there some experimental facts of designing such a protein, or you come to these conclusions only by computational methods? So in real lab, is someone who can design protein?
Yeah. It's a protein design is a very well established field, but and actually to design a stable protein is considered now relatively easy. Or for example, I don't know, maybe you already know it, but when you wash your clothes with like most of the washing powder include now enzymes that were industrially designed to increase stability. So this is something that is commonly done. But to design for function, that's still challenging. So to design for adenine binding, for example, that was not done. So we are in contact with several collaborators who hopefully will try to use the themes to design.
Thank you very much.
Thank you.
If no other questions, maybe we can stop here. What do you think?
Yes, thank you. Thank you very much for inviting me.
Thank you for joining us. I hope we will meet in person.
Yeah, that'll be much better.
Let's say in near future.
Yeah, that'll be much better.
Yeah.
Okay, thank you again.
Thank you again.
Thank you very much for organizing all that, for introducing.
Yeah. Thank you.